HIV is far more pernicious, obviously has spread around the world with very large numbers of people infected. Also has a large sequence diversity, more than HCV, with very different types. This is even just for HIV-1. There's also HIV-2 with geographic distribution of these different clades of HIV, most coming from the M group. And many studies looking at whether or not the different clades of HIV actually cause different types of disease in terms of pathogenicity, transmissibility, and route of infection. So again, another enveloped virus here. We've got the envelope glycoprotein, GP120. This is going to be our entry factor, like HA, like spike. And then we have the viral capsid formed by this matrix protein, which is called GAG in HIV, equivalent to the M protein in flu that we were talking about, or the matrix protein in SARS-CoV-2. And then the RNA molecules, and then a very, very special polymerase that we'll talk about in more detail in a minute, along with several other structural proteins. Sort of like HCV, you have this one long genome, single-stranded RNA virus. You have two copies of the genome, but those are not, they're identical segments. They are not like flu, where you have multiple genes and they're divided between different segments. And so the three major structural proteins are the GAG, which is that matrix, the polymerase, plus the envelope. And that is all done in one frame off of the main genome. And then you have these alternate reading frames that produce all of these other important proteins. So what makes HIV special is that it's a retrovirus. It's a human retrovirus, of which there are not very many. It's an RNA virus that replicates by making a DNA copy of itself. So all the viruses we've talked up to by now are either positive or negative stranded RNA viruses that in their replication cycle make the other strand positive or negative to then template the reverse of that. HIV takes RNA, goes through this reverse transcription process using its very special polymerase, reverse transcriptase, and then integrates that DNA genome into your host cell DNA genome, where it is now then part of your host DNA. And if that cell ever divides, it will propagate the HIV genome with its division. And it will also use that template of the HIV in its own genome to make more HIV virions, exactly as if it was a protein, a normal set of proteins within inside the cell. So one infected cell can become a stable reservoir for the long-term production of viral particles. So this is very different from HCV. Lots of similarities, even to the viruses that we were seeing earlier. Envelope virus finds its receptor. HCV has multiple receptors. HIV has a single primary receptor, which is the CD4 molecule on CD4 T cells. This is another way in which it's particularly pernicious, because it's going to target the cells that are going to be essential for its destruction. So it uses CD4, like SARS-CoV-2 uses ACE2. Very specific binding, uncoding. Again, we have to uncode, we've got to get our genomes out. We got to find a place to replicate. Now we're going back in the nucleus, kind of like flu. Unlike flu, flu always stays RNA, HIV becomes DNA, integrates into the genome. HCV is just hanging out in the cytoplasm and the membranous web. And then both viruses are going to translate new proteins and bud out from there. HCV purely RNA, HIV has this DNA intermediate that allows it to be heritably integrated. HIV works on an even longer time scale than HCV. You have this early acute infection that feels kind of flu-y, high levels of viral replication that are controlled over the course of weeks by your innate and adaptive immune responses, and we'll talk about those here in a second. But like all viruses, intracellular viruses that are going to be targeted by a type 1 response, that's what we're going to be looking at. But then this long clinical latency with low levels of viral production, but constant levels of viral production in the absence of any drug treatment. And this is important. Keep this concept in mind for when we get to herpes viruses. It's not enough to trigger a full-scale inflammatory immune response. This is on the course of years. People aren't going to even know they're feeling very sick, but there's this slow degradation of the viral reservoir because you do have this slow churning immune response that's trying to kill virally infected cells, and those virally infected cells are CD4 T cells. And so you end up with this slow, constant, almost imperceptible decline of the CD4 T cell compartment, which then collapses your adaptive immune response. These are the quarterbacks of the immune system. They can't quarterback anymore. Once they cross a certain threshold, you can't make new immune responses. Any chronic infections that you're controlling with CD4 T cells, like HCV, like TB that we'll talk about tomorrow, are now going to be unrestrained from that type Th1 type control, and you're going to have recrudescence of the HIV itself and the reactivation of a number of other infections. So HIV kills the cells that it's in, these CD4 T cells, and it kills it in a number of different ways. It's a highly productive virus like HCV, so it's putting a lot of stress on the cell because it's stealing resources from the cells. It's stealing its membranes, and it's compromising the ability of the cell to just function as a normal cell because it's spending all of this treasure on making new viruses. Then some of these non-structural proteins in HIV actually activate cell death pathways. And it's not really well understood why HIV wants to kill its cells. I think it's just to cause derangement in the immune system, and I'll show that on the next slide. But HIV actively works to destroy its reservoir at some level. So it's important to keep in mind that this is not just the loss of CD4 T cells, although that is very important, but it's the dysregulation of CD4 T cells. So in addition to them dying, they're also activated as they die. And so if you think about, here's your HIV infected T cell down here, it's hyperactivated because it's dealing with its own internal infection, just like if it was an epithelial cell that was infected by the flu, trying to deal with that, also trying to deal with potentially deal with the ongoing immune response. If it's an HIV specific T cell, how much worse this is for that T cell because it's activated by conventional T cell activation, also has the HIV inside of itself. This is another reason for HIV to actually actively kill its reservoir because its reservoir is also its destroyer. But as part of these two sources of activation, conventional activation and infection-based activation, the CD4 T cells go crazy and start producing wild amounts of inflammatory cytokines and not particularly well-regulated inflammatory cytokines. And so the quarterbacking that the CD4 T cells are doing is poor, and they're going to dysregulate your immune response. And so in addition to being hyper-inflamed, you're also misdirected and you're making bad immune responses. And so your immune response just becomes overall compromised. And as a result of that, you can reactivate all kinds of different infections, things like parasites, toxoplasma, which is an intracellular parasite, intracellular bacteria. Again, we're really hitting at the Th1 targeted responses, these type 1 responses where we need cellular immunity to kill the pathogen inside the cell. And that also includes herpes viruses, which we're going to talk about in a second. These are 3 herpes viruses right here. And you see people activating herpes viruses like EBV, Kaposi sarcoma, and driving these fatal conditions. So I think it should be clear by now why HIV is unlike any other chronic infection, because it's using these traditional immune evasion mechanisms. So it uses things like CTL escape, it mutates itself just like HCV, exactly the same types of experiments can be done with HIV, showing it accumulates mutations over time, antigen masking, just trying to hang out and not cause too much trouble like HCV does, versus in combination with this very non-traditional approach of directly attacking immune function and cell compartment. So getting rid of the CD4 T cells that are going to be necessary for coordinating the overall response. And so you have this massive accumulation of dysfunction and death that then promotes HIV replication. So can we effectively control replication? The successes to date have not been immunologically based, and we'll just briefly touch on that here in a few more slides. The successes have been drug-based. And the drugs have targeted these many unique features of the HIV life cycle. And so that is one way in which viruses that get more and more clever, including herpes viruses, HIV, can be easily targeted compared to something like flu or SARS-CoV-2, where you have not that many things going on, very simple genomes, very simple life cycles in in flu and SARS-CoV-2. And you have the viruses trying to get out of the cells very quickly. So by the time I think I have the flu, I wait a day, I go to my doctor tells me to wait another day, then they finally call in a prescription. The course of my infection is pretty much decided for the flu. So drugs aren't going to do very much, and that's why drugs in general don't do a lot for influenza unless you're in a high-risk group and you get that drug very, very, very quickly, something like Tamiflu. For HIV, you've got years to target these really unique biological pathways that can be exploited. And so things like the integration event, where you're sticking the HIV DNA into the genome, that doesn't happen in a normal cell. So that could easily be targeted by a drug. The specific proteases that allow the HIV virion to mature and cleave those long polypeptides that get made, again, that is a very specific pathway using very specific enzymes that don't occur in a normal cell. And for any kind of drug approach, that's what you're looking for, are steps that are not part of your normal biology. And of course, the reverse transcription event, where you go from RNA to DNA, never happens in a normal eukaryotic cell. And so easily targetable without having a lot of toxicity. So there are multiple types of reverse transcriptase inhibitors. There are those nucleoside analogs, just like we talked about for HCV, like ribavirin, where you use something that looks like a nucleotide to an enzyme, but it's not really a nucleotide, so it stops replication. Or you can have a drug that actually directly binds to the reverse transcriptase protein itself and inhibits it, blocks its active site. And that's called a non-nucleoside reverse transcriptase inhibitor because it doesn't look like a nucleotide. There's the protease inhibitors, which were a real game changer in the mid-90s because they target this unique enzyme that's in HIV. Now, your cell's full of proteases, but they're not full of this specific kind of protease that could be targeted with a very specific drug binding to the active site. And it was a combination, actually, of the protease inhibitor with a nucleoside and a non-nucleoside inhibitor as a triple drug therapy that really led to the great achievements in HIV control in the mid-90s. And again, that was also done through a collaboration with Alan Perelson and math modeling that he developed that argued that you would actually need all three drugs to limit the mutation rate of HIV to get around any single one of those drugs. If you treat someone with any one of these drugs in isolation that are highly effective together, that person acquires resistance. And so, hold on one second, sorry, just making sure that call wasn't an emergency. And so it's the combination of the three that limits the mutational space that the virus can achieve that led to the control of the drugs. There are some more recent drugs that are less commonly used, including the integrase inhibitors and fusion inhibitors that block viral entry. Again, fusion is a key part of any virus, envelope virus getting out of its endosome into the cytoplasm, but most of the drugs now are still protease nuclear RT inhibitors and more commonly now integrase inhibitors. So the problem with HIV in terms of long-term control, why the drugs are great but not the ultimate solution so far, is that you have this lifelong reservoir of the virus, particularly in a cell that's quiescent, that has gone into a resting state, is not activated, maybe is not HIV specific, is not specific for anything in the body. It could be holding on to a silent provirus, the HIV genome in its nucleus, and 10 years from now get activated because it happens to be specific for an antigen that you come across, and that will activate HIV production, and all of a sudden you're back to a full HIV infection. So these, there were many, many trials where people tried to interrupt drug treatment because they figured they'd knocked the levels down low enough, and many of those failed because people could reactivate even after months of quiescence by turning these viruses back on. And that gets into what the individual reservoirs are. So if you take a person and you give them this triple drug therapy, you have, again, just like we saw for HCV, but a very different kinetic. You see these phases of drops, and the amount of viral RNA that you can measure takes about 6 months to get to this very, very low set point. And what you're doing is you're eliminating each of these reservoirs, but the final reservoir that we can't get rid of is this quiescent T cell reservoir, just lurking out there with these HIV genomes in them. And it's why people stay on the drugs, usually at relatively low doses for the rest of their lives. the big focus in the HIV field has been whether or not the immune system can be used to clear infection. And at first, it's a philosophical question: is it even possible, given how clever and resourceful this virus is, that we can get sterilizing immunity? Because that's ultimately what you need. You need sterilizing immunity, maybe through a combination of antibody and CD8 T cell responses, but you've got to clear every last vestige of the virus from your body. And so we know you do make an immune response, and it controls the viral load for many, many years, at least at some level. And these mechanisms of immune protection are the ones that we've been talking about. We've got neutralizing antibodies that are going to bind to that GP120 protein, which is like the spike, which is like the HA, which is what the virus is going to use to get inside the cells. This would be our best chance at sterilizing immunity. And this is where the focus of a lot of HIV vaccination has been, is trying to block the virus from ever getting inside the cell. But as I've tried to keep emphasizing, you've got to get every single one, because if one gets in, you now have an eternal, essentially, reservoir of virus that can be produced. And so the alternative is a combination with antibodies and CD4 and CD8 T cells that can go out and kill infected cells that have snuck through this neutralizing antibody border. So there were a number of experiments in the 90s and 2000s to try to define the role of CD8 T cells, often using the primate models, where if you depleted somebody of their CD8 T cells, you could see very easily, obviously you wouldn't do this to a person, but if you did this to an animal model, you could see very, very high levels of virus in these individuals. So one of the goals of a vaccine program was initially thinking about an ideal vaccine that would just completely eliminate virus. But given the public health burden of HIV, there was some thought that you could have a potentially suboptimal vaccine that wouldn't necessarily block infection, provide sterilizing immunity, but lower this set point, this period of time during the weeks to years, to a level that was well below where people's natural immune response would set it, because we knew that that set point actually correlated very well with how long people stayed healthy and without any obvious consequences from the HIV infection. And so the idea was, maybe it's not zero, but maybe if you get it down low enough, you can significantly control infection. And that has largely been disappointing. Again, I think because of the potency of this reservoir and the fact that any single cell can repopulate it and raise this set point if you don't have a full panoply of good immune responses. The other thought about this was that it would be a way of limiting spread, which has now become less of a focus of vaccination because of the very good pre-exposure drug regimens. So one of the big shifts in HIV vaccination came from an adenovirus-based vaccination program that was led by Merck, that was using a viral vector to try to stimulate both antibody and T cell responses. And it was a mix of different viral glycoproteins. And this was done in the mid-2000s in collaboration with the NIH. And the thing that really shook the HIV vaccine field and what really led to a reevaluation of a lot of these programs, and also came up again in SARS-CoV-2 with the adenovirus vaccines, is they used a very common adenovirus, adenovirus 5. And this is different than what Johnson & Johnson used in SARS-CoV-2, which was Ad26. Lots of people had antibodies to adenovirus 5 because they'd been infected with adenovirus 5, just naturally. It's a cold virus, you wouldn't maybe even know you had it. So they looked at the results of the study, and they stratified people based on whether or not at the beginning of the study they had antibodies to Ad5. So these people had no antibodies to Ad5, these people had sort of moderate levels, these people had high levels, these people had very high levels. Once you got out of the group that had very low levels of adenovirus 5, people that got the vaccine got more HIV than the people who didn't get the vaccine. So this is kind of like the RSV story I told you earlier, where if you had antibodies already to adenovirus 5, it seemed to actually generate a kind of immune response that made you more susceptible to HIV than if you didn't get the vaccine. So this was a disaster, because you can't ask people to go into a vaccine regimen where there's even a chance that you're going to end up more likely to get infected. And so this led to the cancellation of many, many vaccine trials. It led to the cancellation of a lot of the focus on cellular immunity. And this, I think, was really the shift towards the real big focus on antibody immunity, which has also been hugely disappointing, even as recently as last year. And I can talk about that more in the questions if people are interested. There was one vaccine trial that has shown some promise, and that's actually driven a lot of the antibody immune-based focus. And this was this RV144 trial, and you can see the curves here. So if you've got the placebo versus the vaccine, you can see there's a real difference in the number of infections people are getting in the vaccine group. Nothing like the really great vaccines we've come to know and love, but maybe a 30% reduction. In people that got infected, they did not do any better or worse. And so it seemed to change the rate at which you got infected, but then once you were infected, you had the same course as everyone else. And then they looked at what actually was a correlate of getting HIV, and they found the statistically significant differences were in antibody measures, specifically in antibodies that were associated with a binding to V1, V2 loops of the envelope protein. And so it's hard to see that difference there, but there is a difference there in that people that went on to not get infected had higher levels of those antibodies. Okay, so what I've shown you in this part is that HIV is really a unique pathogen in that it targets the immune system directly. In a way, it's of all the viruses and pathogens we're going to talk about, it's playing the most offense of any of the pathogens we deal with, because it's dysregulating the cells at the heart of coordinating all of our adaptive immune response. Antibody and cellular immunity all have to flow through CD4 T cells. And so to have this pathogen that's exquisitely focused on dysregulating and killing CD4 T cells makes it really an ideal pathogen. And despite this really ideal pathogen, your immune response is relatively resilient for the initial phases of the infection. Antibody and CTLs provide that early control, keep people alive for 8 to 10 years. And then it's that loss, that slow degradation of immune competence that then drives the pathogenesis that we see. And so far, drugs are really still the most effective treatment tool. And like I said, there has been a lot of work in the last couple of years on broadly neutralizing antibodies, which we can talk about, which were a big hope, and I think have been largely disappointing.